Well, thank you all for inviting me to speak today. I feel honored to be on this list of kelp specialists, kelp enthusiasts. So I'm going to talk about two projects that cover the themes of kelp thermal tolerance and genetic diversity and how these interact with restoration success. This has been the focus in these of my postdoctoral studies. And I'm going to be focusing on these two projects that were from my first year as a postdoc, 2023, co-supervised by Julia Baum and Christopher Neufeld. So we've had many talks today that mentioned kelp is declining largely due to temperature. That's one of the stresses people are noticing is coming out as on top. This is a figure produced by some of my colleagues in a recent paper showing areas where kelp has been lost, been stable, and gained. So we see a lot of these areas of loss are concentrated around these inland waterways where temperatures are already reaching kelp's thermal limits. So I am interested in ecology at large. So the key questions in restoration ecology that I work to address are, can we use local adaptation to improve restoration outcomes? And how does genetic diversity influence restoration success? And these are very commonly studied questions in the plant restoration literature, but we have sort of not had as many years looking at kelp as the plant world has. So classically to detect local adaptation, people do what is called a reciprocal transplant. And this is the idea of taking material like a plant or an animal, some species, and swapping it from its home site to a different site and also outplanting it at its home site and doing that vice versa. And you would expect to see that the organism in question would perform better by some metric at its home site than an away site if it is locally adapted, and vice versa. But we might also see an ecotype effect. So this is when one genetic group just does better overall, regardless of where it's planted. And you might also see a site effect. So some sites are just prohibitive of growth, and others, the species can grow fine. So the hypotheses that my work has addressed is that first, kelp will perform, or these are sort of expectations, is that kelp will perform best closest to its local site, and this is the local is best hypothesis. Alternatively, we might see that kelp from a cool site survives and grows better overall, regardless of the outplant site. And this would happen if that kelp still has higher genetic diversity and hasn't experienced as many stresses. For example, we might be getting bottlenecks and population decrease in the really stressed warm sites. So finally, that ties into the genetic questions, and I would expect cultures with low genetic diversity will survive and grow more poorly. So I'm focusing this talk today on two projects by the Kelp Rescue Initiative in some of our focal areas. The first one is in Barkley Sound. So this is on the west coast of Vancouver Island. We've had many talks about this area today. And this is where we'll be focusing on genetic diversity and local adaptation. Barkley Sound is a coastal waterway. We see there's a strong temperature gradient that has only been exacerbated by the blob marine heat wave. So this figure, also by Starko et al., many of my colleagues, is showing on the top the thermal gradient along the sound getting about 3 degrees warmer as you go inland. And the dots are showing kelp populations that either were stable in blue or were lost in red after the marine heat wave in 2014 to 2016. So in this survey, about 43% of sites lost their kelp. So in this study, we wanted to address our local populations that persisted through this marine heat wave more heat tolerant. So we're really looking at this question at a population level. We're not yet honing in on some of those strains like some of the other speakers today have been looking at. For this experiment, we chose a cool site, Ed King Island, which has been sort of on this exterior cooler area with less kelp loss, and then a warmer site, Dixon, that still had a tiny remaining population, but most of the kelp in this area has been lost. And we did this reciprocal transplant, like swapping some of the kelp between these sites. I just want to point out that the cool site had cooler temperatures, higher nutrients, and more urchins, and the warmer site had lower nutrients and fewer urchins. We are using the green gravel method for this experiment, growing a baby giant kelp. I should mention that this study focuses on giant kelp. And we're growing them in these separate little bins so we can do different treatments to outplant in a factorial experiment for this study. So these are the treatments we focused on. We had our parent kelp that had come from cool and warm populations as two of the treatments. And then we also were interested in this question of genetic diversity, but we wanted to manipulate it directly by having some of the cultures have only 2 parent kelp and some have 10. And we outplanted this 8 times at each site. So this is some photos of me and Clay placing these baby kelps out by hand. It was very cold. This was in like February and there was snow on the boat. So this is an example of the fact that, look, our kelp actually grew. So this is a cute baby kelp that has, you can see some of its floats developing, which we're very excited about. You can tell by our hand motions. So there's a lot of results from this study. This is a under review manuscript, but I'm just gonna, for brevity, go through survival. So this figure is showing sort of our reciprocal design. We have the warm and cool outplant sites. The colors are the warm and cool source populations. And I'm showing the percent gravel remaining with kelp over the 3 time periods, which are those separate panels. So now to look for evidence of local adaptation, we'd want to see a significant interaction between the source population and the outplant site, which we do at time point 1. That shows a significant interaction. But what is interesting is we see the expected result with the cool population, where it's growing better at its home site. But for the warm population, it was also growing better at the cool site. So that might be a sign of maladaptation in that population. At later time points, we didn't see this interaction. But looking at the number of parents, we saw an interaction between parent number and source population, which was interesting. But we also saw, in general, the cool population grew slightly better overall. This was like a pretty small effect. The same sort of patterns came out for growth. I'm not showing this today, but with growth, we also saw that site had a significant impact at the 3rd time point, with kelp growing better at the cool site, possibly because it has higher nutrients as well. We also looked at genetic diversity. So these are for the parent kelps and some of the offspring from culture used in this experiment. Here we see observed heterozygosity as a metric of genetic diversity, and each point is representing a kelp individual. So the first thing you notice is there's a fundamental difference in diversity level just with these 2 populations, the warm and the cool. The cool population had higher genetic diversity overall. We saw this expected result where the 2 parent cultures had lower genetic diversity, which is great, our treatment worked. But what's really interesting is the reason the average diversity is lower is some of these kelps have about half the heterozygosity of the others, and that's bringing down the mean. That is actually a sign that these are produced by self-fertilization. So we saw 50% selfing rates in the cultures that were produced with 2 parents. Another interesting observation, and we didn't have the sample size to test to get significance from this, but we did observe that the kelp that were selfed had a higher mortality rate by the end of the season. So moving right along, our next case study that I wanted to highlight was looking at a very warm region, Hornby and Denman Island, and this is with bull kelp. So this is a case where we want to start tackling that Salish Sea question of, can we restore in the Salish Sea by finding populations that perform better? So in this experiment, we sourced populations from a lot of these still existing populations along this thermal gradient, many of which have been talked about earlier today. And we outplanted them in a common outplant site. This is called a common garden experiment. And this outplant site was at Hornby Island, which is where that star is, right in the middle of that super warm range. This was in a partnership with Hornby Island Diving. Rob and Amanda Zielinski have been stewarding this seeded line array for a decade, and they wanted to partner with KRI to make an experiment out of it and get some useful information. So we grew these 5 populations on these floating lines, helps us track them really easily. And we had replicate lines for each population that were randomly distributed so that the position in the array is not affecting our results. All right, so this is showing the bottom temperature at the site throughout the time series, and I'm going to superimpose our monitoring and sort of steady time points. So we put them out in January, then we took random stipe length measurements in each population at 70 days, at 120 days, and then once the water had reached peak warmth at 140 days. So at this point, we also put a hole punch in the blade so that we could go back 10 days later and measure how far it had grown so we could get at blade elongation rates during the maximal stressful period. I also want to point out that I've superimposed lines at sort of 11-ish degree, 12-ish degrees, and 18 degrees at different levels where we've known kelp like to grow, and the red is showing where kelp really are unhappy. So we're starting to see temperatures like going into really stressful range here, which is great. That's the goal of this experiment, is to really stress them out. So once again, we had a lot of results from this study, but I wanted to just show what I think is one of the most interesting figures. These are bars showing blade elongation rates, that's growth per day, in the different populations. And I've ordered these populations by distance from the source site to the outplant site. The closest site to the outplant site, Oyster River, had huge blade elongation in this super warm period when a lot of the other populations were having their blades just crumble off and be completely covered with bryozoans. So we're seeing significant differences, especially the Victoria, that's the coldest and farthest population, was unhappy. We see some different results for stipe length. What was interesting is the Browns Bay population actually had the longest stipe length, so really good stipe growth. The Oyster River population had pretty short stipes overall. So this could be seen as a bad thing. This could be they're stressed and their biomass is lower, but it also could be yet another adaptation where possibly they're keeping that stipe length low to remain below the thermocline and not be as, not access these hot waters as much. So key takeaways, it's sort of hard to synthesize this whole talk because they're two very different systems, very different study areas. But what we seem to see is that in Barkley Sound with giant kelp, this was a small spatial scale, so looking at microclimates in a single sound, whether there would be local adaptation there. And we didn't see very strong evidence. We might have seen a little bit in the cool population, but what we really saw was the warm site might actually be a little maladapted, and this could have been because it had such population loss during the most recent stressful event. At larger spatial scales, so this was the entire Strait of Georgia, we saw that local populations performed best. But there's some nuance there because we were looking at stipe growth and blade elongation and persistence and reproduction. So I'm definitely happy to talk about more of these results. And it's sort of hard to say at this point what's best, but I think there might be a scenario where mixing some of these populations could confer mixtures of different useful traits. Generally, the population that had higher genetic diversity performed better, but once again, this was a pretty small scale limited study, so having more experiments doing this kind of question would be super useful. And then self-fertilization rates were high in cultures that had few parents, which we already know, but it hasn't actually been quantified how much selfing would really happen in these cultures. So this is yet another sort of warning to use higher parent numbers when you're culturing kelp. So we have many funders and sponsors, and I thank all of them, and I'm happy to take any questions.